Hi and welcome to our presentation on conservation genetics. Science in the service of nature. We are the Golgi gang, Akala, Afika, and Tayrunzie. Let's learn how genetics helps save species and protect nature. Conservation genetics uses science to protect animals, plants, and ecosystems. It studies how species survive and adapt to changes in their environment. This field combines genetics and biology to support conservation efforts. This field covers many areas, predicting risks like habitat loss, studying how genes help species adapt, looking at diversity within populations, restoring ecosystems, and reintroducing species into the wild. Scientists use tools like DNA sequencing to study genes, environmental DNA to find species, and molecular markers to see how populations are connected. They also use computer models to predict risks and changes. We will explore how genetic diversity affects species survival and the implications for conservation strategies. Charles Darwin first recognized the importance of genetic diversity in 1896. Recent advancements in biology have underscored its critical role in enabling species to adapt and thrive in changing environments. While protecting habitats is essential, maintaining genetic diversity is equally crucial for long-term survival. Small or endangered populations often exhibit lower genetic diversity due to population declines. This lack of diversity makes them more vulnerable to extinction. Understanding the mechanisms that lead to this vulnerability is vital for effective conservation. Inbreeding occurs when closely related individuals mate, resulting in reduced genetic variability. This leads to inbreeding depression, where inbred individuals typically have lower fitness compared to those from diverse genetic backgrounds. Such reductions can contribute significantly to population decline. Inbred individuals face a higher risk of extinction due to increased homozygosity, which reduces their overall fitness. This slide highlights how fitness impacts are pronounced in small populations where the loss of heterozygosity can diminish survival rates. To mitigate the effects of inbreeding depression, introducing new genetic diversity from other populations can be beneficial. This strategy helps enhance fitness levels and improve the chances of survival for endangered species. Natural selection plays a critical role in maintaining genetic health within populations. While it removes harmful alleles, new mutations can appear faster than they can be eliminated, particularly in small populations. This dynamic creates a balance that is essential for population sustainability. The extinction vortex represents a destructive feedback loop where small populations decline, leading to reduced fitness and increased vulnerability to random environmental events. Understanding this process is key to developing effective conservation strategies. Both biotic and abiotic factors contribute to the extinction vortex. Biotic factors include genetic deterioration and reduced reproduction, while abiotic factors encompass environmental changes that further threaten small populations. The extinction vortex operates through several mechanisms. A decline in population size leads to reduced fitness, which accelerates the decline toward extinction. As populations shrink, random fluctuations become increasingly significant, often overshadowing initial causes of decline. To prevent extinction, conservation efforts must prioritize maintaining large and genetically diverse populations. This includes understanding the concept of minimum viable population, MVP, which is the smallest size required for a species' long-term survival.
Scientists have learned a lot about genetics, but there's a big gap between what we know and how we use it to protect species. This conservation genetic gap means many animals and plants are still at risk. For example, we know genetic diversity helps species survive, but many conservation plans don't include this. To fix this, scientists and conservationists need to work together better. By using genetic knowledge in conservation, we can save more species before it's too late. Conservation often focuses on management units and use specific groups of animals and plants that are independent but still connected genetically. Protecting MUs help to keep populations stable and reduce pressure on small, vulnerable groups. For example, the Florida panther was saved by adding new genes from Texas cougars. This shows how important MUs are, but remember, MUs are just one part of the solutions. We also need to protect entire habitats to ensure the species survive in the long term. Evolutionarily significant units, ESUs, are groups of animals and plants with unique evolutionary histories. Protecting ESUs helps keep genetic diversity high, so species can adapt to future changes. For example, saving different groups of salmon in the Pacific Northwest has helped them survive climate change. But ESUs are different from MUs. MUs focus on short-term survival, while ESU aim to protect long-term evolution. Understanding this helps us create better conservation plans. Genomics, the study of entire genomes, is changing how we protect species. It helps us find genes that help animals and plants adapt, track population changes, and even learn about the species' past. For example, scientists use genomics to discover that the Libran lynx survived a genetic crisis 10,000 years ago. This knowledge is helping save the lynx from the extinction today. But genomics isn't a magic fix. We still need to protect habitats and use other conservation methods to save species. Ecogenomics takes genomics a step further by studying genes in their natural habitats. It helps us understand how species adapt to their environments so we can create better conservation plans. For example, ecogenomics has been used to study coral reefs, finding genes that help corals survive warmer oceans, but ecogenomics is still new. We need more research to fully understand how genes and ecosystem work together, especially as the environment changes quickly. In conclusion, conservation genetics combines genetics and ecology to protect endangered species and preserve biodiversity. Using tools like DNA sequencing and genomics, scientists study genetic diversity and population structures to predict threats like habitat destructions and species mixing. Key challenges include interbreeding, depressions, mutation meltdown, and the extinction vortex, which threaten the small populations by reducing genetic diversity and fitness. To combat this, conservation effort must maintain a large genetically diverse population and establish minimal viable populations. Advancement in genomics and ecogenomics can transform conservation, offering insight into species' adaptive potential and historical population dynamics. For example, genomics saved the Libran lynx and Florida panther by reintroducing genetic diversity. However, a gap remains between genetic knowledge and its application in conservation. Closing this gap requires stronger collaborations to integrate genetic insight into habitat restorations and species management. Concepts like management units and evolutionary significant units emphasize protecting both short-term survival and long-term evolutionary potential. By combining genetic research with ecosystem-level efforts, we can address challenges like interbreeding and population decline, ensuring the species resilient in the changing world.